everybody to be here today and to attend this fourth workshop on data storage on DNA. So we have today a nice program with three speakers. The first one is Ogika Milenkovic. She's a professor at the University of Illinois at Urbana-Champaign. So she's developing new approaches for studying problems of in bioinformatics and bioengineering using coding and information theory. So she will give a presentation today about compression, learning, and error correction for DNA-based image storage. The second talk is given by Nimesh Pinama-Neni. Nimesh is co-founder of ElixWork Technologies. And ElixWork is a biotechnology startup that makes synthetic DNA and uses secure DNA storage to verify food and pharmaceutical for blockchain. So Nimesh will present today a talk on writing data to DNA, what breakthroughs are needed to make this technology feasible. And the last talk is given by Marthe Collot. So Marthe is has a PhD in molecular biology, and she is an engineer with an experience of more than 15 years at Imagen as R&D project manager and operation manager. So she will give a talk. The title of our talk will be Imagen Technology for Ambient Temperature Storage of DNA. So after this free presentation, we have a session of question and answer. So if you have questions during the presentation, please feel free to write them in the chat. So in the chat window, so we can then ask at the end in the wrap-up session, ask the question to the speakers. So I think I don't forget something. So maybe we can start. It's time to start. So Ogika is starting with a presentation on compression learning and error correction for DNA-based image storage. So the floor is yours, Ogika.
Yeah, thank you very much, Mark, for the introduction. Let me see if the oh, it says I cannot share the screen while other participants are sharing. So
okay, sorry.
Can you just, there we go. Great, thank you. Okay, so I hope this is visible. And that enter full screen will work. There we go, and I'll just remove this. And I know the talks are 25 minutes, so we're a bit pressed with time as usual. So first I would like to thank the organizers, Mark and Turaj, for the kind invitation. And I just want to mention that I slightly changed the order of the words and the title. It's going to be Compression, Error Correction, and Learning for DNA-Based Image Storage. This work is done by 3 students of mine and some collaborators here at UIUC. And you will see the last name Tabatabai coming up quite a lot. It's 2 different people that worked on this project with the same last name. So let me start with a very, very brief overview. I'm assuming everyone here on the call knows about DNA storage. And I'm just going to give credit to the 2 groups that started the whole area in 2012 and 2013, Church and collaborators and Goldman and collaborators. And the idea behind this new molecular storage platform was really something that came about due to deep considerations of what DNA has to offer as a storage media. It's obviously ultra large storage media because we can squeeze in 2 bits of information in roughly 0.3 nanometers of length. It's highly durable, as we all know, but it's also a universal storage media, which is something to bear in mind because many of you may remember floppy disks, magnetic tapes, punch cards that I will show in some of the pictures. These are storage technologies of the past for which we have pretty much no interest anymore to put them in use, because we have moved along with disks, with flash memories and other devices. And it's very hard to find systems nowadays that can read content from floppy disks, for example. While if you store information in DNA, then you're guaranteed that in many, many decades and centuries to come, you will be able to read the content stored, because we have an insatiable appetite to read DNA, mostly for biomedical applications. So what Church and Goldman proposed was a very straightforward conceptually scheme, and very original, obviously, at that time. And it involved this diagram that I'm showing here that asserts that you should just take digital content, convert binary content, for example, encoded into a 4-letter alphabet with some tiny additions to it, something like error detection, parity checks. Then once you encode the information in DNA, all you have to do is call your favorite DNA synthesis company, IDT in the case of our group, or Twist, Agilent, and others. You will get the content that you wanted to store encoded in DNA. And then when you want to read the content that you stored in the molecules, you just have to use, again, one of your favorite sequencing platforms, either Illumina, Oxford Nanopore, or some other device. And then you have to revert back from a quaternary alphabet to a binary alphabet, back to the original data that you stored. So that all seems very doable. And there is absolutely no doubt that this is a technology that started off with something that is already easy to do, but the issue is the price tag that comes with it. So this is a table we created a few months back, showing how much do you have to pay to different companies to get a certain amount of DNA. And this red block, if it doesn't make you nervous already, should make you nervous right now, seeing that if you want, let's say, 600,000 oligos in very small concentration, length 120, you have to pay $60,000. IDT, for example, for length 20, which is 40 bits, asks for roughly $7.5. So these are very high prices, and this has been really the bottleneck, and I'm pretty sure you're going to hear more about that in the talks to come, for all the work that is being done on DNA storage, the issue with the price. And that obviously brought up the topic of doing compression, because compression is one way to save in terms of storage costs, because you don't store the data in raw format, but you compress it. And once it's compressed, the fingerprint or the storage footprint of the data should become smaller. So it's not surprising that, in particular, Church and Goldman, the first two people to come up with this concept, proposed using compression. And here's one of the pictures, because I'm going to focus mostly on images now in this talk. This is one of the images that was stored by the European Bioinformatics Institute team. It was in JPEG 2000 format. And they used, as pointed out here, JPEG 2000 for image compression. And a lot of other works that I will not have time to talk about address similar issues, basically how to do image compression so that it nicely fits into this DNA storage paradigm. But the problem was, even with the work that Church and Goldman in this case did, is that they had reconstruction errors. And basically, I denoted these as two blocks that you can easily see. These are not exactly the errors that you saw in their work, but I just drew these blocks to explain what kind of errors you can see. You can see errors of this type, which are basically due to the fact that some oligos are missing. And it really depends on the company that you use to synthesize your DNA, how big of a percentage of missing oligos you have, or oligos corrupted to the point that they cannot be used. So that usually manifests itself as missing oligos that appear as black blotches in the DNA. Then you can have errors, sporadic errors in the oligos that you use that create discolorations, for example, like this little cloud that I added in the tree. So you have to be careful that when you do compression, all these errors that occur naturally both during the synthesis and sequencing process have a tendency to propagate. And catastrophic error propagation is a big issue. So you need to couple compression with error correction and other restoration or reconstruction methods, in particular when you deal with images. So what I will be focusing on throughout the remainder of the talk is two parts. Part one, which discusses joint source channel coding, so basically how to do joint compression and error correction. And then the second part will focus on what I will call joint source coding and learning, because as you will see, channel coding adds redundancy, which amounts to additional cost issues with the storage platform, and that's something that we want to avoid. And that motivates the use of machine learning techniques that are emerging left and right pretty much with the craze we have in machine learning and CVPR research. I will discuss how to use natural redundancy in images to try to combat the effects of synthesis and sequencing errors. So let me start with the first part, joint source channel coding. So for this purpose, I will just go over a fairly old paper of ours, 4 years, if you want to call that old, that discussed what I believe is the first joint source channel coding scheme for storage. And it focused more on the channel coding part because there were not really too many innovations on the source coding part. So what we did is the following. So we took raw images, let's say a smiley face, where you will see some images of Citizen Kane, and we just ran simple JPEG compression on the images. And then we did base64 conversion and then some form of encoding. And I added a cross here to say, let's just assume that encoding amounted to nothing else than converting binary data into a quaternary alphabet ATGC. And in most of our prior work, we really didn't use oligo pools. We use G blocks that are of larger length, roughly 1,000 or 2,000, as you see in this picture. And we also used addressing schemes or labels or barcodes for the DNA for the simple reason that we wanted to enable random access. And that was another paper of ours from 2015. And so when you do a simple encoding process like this, you would like to be able to read the content. And the punchline of this work was that if you want to make the system operational and widely used, you can't require people to read the content with Illumina sequencing machines. Why? Because Illumina devices are extremely expensive, they're bulky. You can't really take them in your purse or in your laptop case. So we focused on reading the content stored in DNA with nanopores. And again, I believe this was the first work that tried to advertise the use of Oxford nanopores, in this case in particular, for reading the content in DNA. And since I don't have enough time, I'm just giving you a graphic illustration here of how Oxford nanopore and other nanopore systems work. You push your DNA through a pore, you observe the drop in the base current, and based on the amplitude and the width basically of the, and the width of the drop period or the event period, you decide if it was an A, T, or G, or C symbol. So this is the scheme that we wanted to use. And it turns out if you just ignore this block, you do direct encoding into DNA without any error correction coding with the JPEG compression algorithm used for each image. These are the kind of results you would get. And this was an early work when we basically didn't have enough money to buy decent color images or pay for synthesis of color image. So we basically used old black and white images like this one from Citizen Kane. And this is the result you got when you read out your content using Oxford nanopores, because there were obviously errors there. Nanopore technologies are fairly prone to introducing indel and substitution errors, and the images obviously do not show any of the details. If maybe if I tilt the screen a little bit, you could see some white contours here, but the image is completely corrupted. So what you have to do is really, if you're going to use any compression method, you need to add redundancy in the encoding domain. And basically, I will explain what kind of redundancy and what kind of bioinformatics approaches went into consideration here. So what happens with Oxford nanopores, you get a bunch of reads that all contain insertions and deletions compared to the original G blocks of length roughly 1,000. And since the G blocks come with address sequences, we did something that's very common in the communications theory literature, wireless communications theory literature. And it was completely unknown at that time as an approach for DNA-based data storage. We looked at these address sequences as pilot sequences that can indicate how good of a quality read we got. So basically, we know beforehand what the addresses are. That's something that we need to know beforehand. And if we see some reads that have the exact address or an address that matches exactly one of the addresses we know should be present, then we can deem that read as sufficiently accurate, a read that is of high quality, maybe a read that passed through nanopores early on when the nanopores were not quote unquote tired. And then once we select only those reads that have perfectly preserved addresses, assuming that these are high quality reads, we ran something called multiple sequence alignment. And that is used to create a consensus sequence. And once you have a consensus sequence, you can try to check if the error correction redundancy, that I will describe a little bit later as well, is allowing you to completely reconstruct the image from the consensus sequence with error correction. And if the procedure didn't work, then you would go through a loop, through an iteration, and try to recruit more sequences that have addresses that have very small number of errors to improve the results and the quality of the images you have. So this is what's going on in a nutshell. And these are the kind of results you get. So with these two images, with the approach described here, without iterating to correct errors, without error correction, you would get the images that I showed before, something like this. But then when you do the selection of the strings based on their quality of addresses, using them as pilot sequences that can tell you something about the quality of the channel, and with error correction redundancy, you restore the images completely back to normal. And the coding schemes that we had involved balancing codes, blocks, balancing blocks of length 8 in terms of their GC content. And these naturally introduce run length constraints, which are also very relevant for the quality of synthesis, and they actually make synthesis easier. We introduced something called asymmetric homopolymer codes to deal with residual errors left. And we toyed with the idea of using repeat-free codes, which will remove repeats that are really dangerous during the synthesis process and can create trouble insofar that you cannot even get the strings synthesized properly. But one question you may ask is, how much did this redundancy here really cost us? If you go back to this diagram here, you see that we try to rely mostly on the fact that every readout platform will give you multiple traces or multiple reads of the same string. The errors we assume are introduced in an IID fashion and independently in each of the oligos. So this whole multiple sequence alignment and read selection based on the quality of address helped a lot, but we still had to use some error correction because of catastrophic error propagation. And the amount of redundancy that we had to add was roughly 0.28 per 2 bits. So the rate in bits per base pair was 172. Out of this 172, roughly 20% of, roughly 0.2 loss can be attributed to error correction. So if you think about it, a 0.2, that's a 10% loss in the density or in the storage content, because you're devoting 0.2 out of 2, sorry, that's 0.2, right. It's a 10% loss, if I'm not mistaken. You're devoting 10% to redundancy that can completely ruin your account, because even 10% can amount to thousands of dollars, especially if you want to store high quality images that are encoded in the RGB system. So that made us think is, can we really get rid of error correction coding redundancy, or any redundancy, especially when we're dealing with images that will contribute to the increase in the cost. So that brings me to the second part of the talk, in where I introduced a new notion, I would call it, of joint source channel coding, source coding and machine learning. And the idea is as follows. So let's try to do some kind of innovative, or let's say just partly innovative encoding or compression of color-coded images, or color images like this Marlon Brando movie, Marlon Brando poster from The And let's see if we can trade off the level of compression to the point that the images look corrupted, but can hopefully be beautified and restored back to a very acceptable visual form using machine learning techniques, so that even when we have some errors, we can correct them using some of the remaining natural redundancy in the images and new machine learning techniques, and not resort to the use of expensive error correction. And there are many other reasons why you wouldn't use error correction. As I mentioned in the previous slides, we used G blocks, which have very few errors compared to oligos. When you have pools of oligos, sometimes up to 30% of the oligos are missing. So our errors that we dealt with were mostly due to nanopore sequencing and not due to synthesis. But you have to bear in mind that during the synthesis process, especially if you use O pools, like the product from IDT or some pools from Twist and Agilent, you will see many missing oligos, which contribute further to significant image distortion. So what did we do? We took a color image and we decomposed it into the red, green, and blue channel. And then we ran, I would say, some standard combinations of compression techniques that have been out there for the longest time. It's the combination that matters. And what we also did is perform a very aggressive quantization of the images. Then we encoded the images, in this case, into shorter blocks. Now we have blocks of length 200, so we have pools of oligos. And the first question we had is, if you have aggressively quantized and compressed images, how should you proceed with adding or using machine learning techniques without error correction to recover from missing oligo effects or from other synthesis and sequencing errors? So the one observation that I should make before proceeding with the next picture is that it's absolutely crucial that you do a decomposition into red, green, and blue channels, and then perform compression on all these three color channels independently, which is okay. People have done that in the past, but it's also extremely important that the red channel, green channel, and blue channel are encoded in oligos, different oligos, and that these oligos are reshuffled when placing the order, because if you use, for example, microarray technologies for synthesis, there are the boundary effects. The oligos that you're synthesizing at spots that are at the boundary of the array usually may be missing or have more errors. So reshuffling the order in which you want the sequence content to be synthesized also matters. But the most important part is to make sure that you do not put information from both the red, green, or blue channel on the same oligo. And the reason for that is very straightforward. As an error control coding person, you immediately think, I have three color channels. I have red, green, and blue. So if I have three channels, that immediately makes me think of a repetition code. Because if I repeat something 3 times, I can always look at the majority call and decide, was it a 0 or a 1, or was it a specific color intensity or not. And if you think about it, let's say this is the image, and I will show more results in a second, that we got of Marlon Brando. If you look at the RGB channels, because we used very different oligos placed randomly in the list for the placement on microarrays, you will see that if you have errors in one channel, for example, in the red channel, I don't know if you can see it, there is a blotch on Marlon Brando's forehead, but in the green channel and blue channel, there is no blotch on his forehead. Here, there is a huge mark on his upper lip, nothing here. Here, just a little bit. So what you can do is, and I'm not going obviously to discuss the details, is you can automatically figure out where the errors happened by just looking at the different RGB channels and the smoothness of the pixel intensity levels in one versus the other 2 channels. So basically, if I were to show you this picture, you would immediately say, okay, there can be some discoloration in this area, but I should be able to tell that it was in the red channel because I have a very smooth forehead in green and very smooth forehead in blue. So once you do that, you can zoom in to the regions that you think contain massive amounts of errors or contain discolorations due to missing oligos. And then you can whiten these locations out. So you can remove those parts that you think are probably erroneous. And clearly in this process, you can make additional errors. And now you're presented with an image that has parts of it missing. And that is the best thing that can happen to you if you work in computer vision, because there has been a flurry of interest in a technique called image inpainting. And that is a technique based on deep learning methods that allows you to fill in gaps in missing images such that you can reconstruct them back to the original or close to original quality. So what we did is we used one possible approach that worked best for us. It's called Edge Connect. And again, due to lack of time, I won't show you how this method works exactly, but it gives you a fairly decent output result. So here are some pictures of Marlon Brando. And we had a huge library of his images encoded. And this is the original, the first row is the original images. The second row is our highly quantized images. Basically, it's probably not that obvious to see on your screen, but we did very aggressive quantization. Visually, it still looks pretty good. The third row contains the reconstructed images with errors. In particular, this image here was really highly corrupted. It's from Streetcar Named Desire. And in this image here, you see a lot of discolorations due to errors that occurred in the synthesis process, actually. And the last row shows you how image inpainting would work. So you still see that Marlon Brando, this image doesn't look quite right compared to this image here because there is a mark on the lip. In this case, Last Tango in Paris, you see a big black blotch on Marlon Brando's cheek. But most images look pretty acceptable when you do this approach, when you use this approach. So we tried other techniques. We were really not happy with just looking at this bottom row because the image looks a little bit washed out. So as I mentioned, there is a flurry of interest in image denoising, image smoothing, image inpainting in the computer vision community. And just last year, there was a really interesting paper called Bringing Old Photos Back to Life. It won the Best Paper Award, I believe, in CVPR 2020. And they show how you can get rid of wrinkles and automatically correct a bunch of issues with the photos that you have. So we did that along with smoothing, and we still didn't get results that were satisfactory. And the issue is that if you do whitening out, if you run our automatic discoloration detection algorithm, you will still see too large of an area where you zeroed out or whitened out the pixel content. And that is especially unpleasant if it happens somewhere in the facial feature area. So what we did is we decided to do something that we call hybrid coding. We only looked, or unequal error protection coding, we only looked at the eyes, nose, and mouth features of each image if they were present. And we added very limited redundancy, only 7% redundancy for the important facial features, which you cannot really smooth out or try to fix using the CVPR method that I mentioned before. And the results look immediately much, much better. So again, these are the corrupted images that we got from the sequencing result. And the last row now shows you the images we got with the extra 7% redundancy only for the facial features and the extra smoothing that we used based on the CVPR paper method. And as you can tell, the images look very, very much like the originals, and I couldn't squeeze the original on the slide here, but you can read more about the whole procedure in this paper that is currently under review by NatureCom. So with that, I just have a few more minutes left, I guess. So I want to mention that you can do much more, even with compressed images, with images that have error correction coding or machine learning reconstruction methods. You can do what you are used to doing with images in the digital domain as well. So we have a completely new line of work that tries to store images with metadata that can be rewritten and with watermarks. And so just to tell you what these two concepts mean in a nutshell, if you think about DNA molecules, there are molecules that have multiple information dimensions. One information dimension is obviously the content, the way in which you represent information traditionally using ATGC. But you can also store information in the sugar phosphate backbone. And there has been work by the Kaiser group and work from our group that appeared last year. And it was featured in Scientific American and a bunch of other publications showing that you can store information in the sugar phosphate backbone by punching it using CRISPR-Cas9 or some other enzymes such as PFAGO. And the positions where you punch the DNA will tell you if it was a 0 or a 1 at that location, or even 2 if you use both strands. And we call this system the DNA punch card system. And what we showed is that those images of Marlon Brando that you had, you can superimpose some metadata about those images in the sugar phosphate backbone by using this concept of punch cards. So you can write metadata, let's say Marlon Brando image or the name of the movie or the year when the movie was made, by punching holes in the sugar phosphate backbone of all the oligos. And then when you're not happy with the metadata you have, you want to erase it. And that's something that's very unique to this scheme. All you have to do is use T4 DNA ligase to seal off the nicks, and the information metadata is completely gone. And then you can do rewriting again, and you can do it in many cycles. And what is really important is that when you do this writing, erasure, rewriting by sealing the nicks or creating nicks or holes in the DNA sugar phosphate backbone, you're basically introducing additional errors in the images, because sometimes the way these nicks are added, some other base may fall off, and then you have additional losses in the strings. And the results I showed you actually account for that, and you have additional errors that we managed to correct with the machine learning method that are due to this metadata writing process. And here is an illustration. For example, on the Godfather image, we stored ownership information such as Illinois. We raised it and recorded a new metadata information called Granger, because we are the Granger College of Engineering. And one thing that I'm not going to discuss because I'm at the end of the talk anyhow, and it's not published work yet, is how to add watermarks to images that I just showed you. And this is something we're just completing. The idea is, again, we can use the topological dimension, which is a dimension that you can view as orthogonal to the sequence dimension. And then you can use the ideas we had about nicking and capping nicks or extending nicks by adding additional tails of DNA, which I'm not going to describe in detail. But these tails, based on where they're added, can encode non-destructive ownership information or watermarks. And that's a really important topic right now, because many of you may know that Google, maybe a few years back, published again a highly cited paper that says that none of the existing watermarks are resilient to attacks by attacks that use deep learning techniques. So they proposed new types for image watermarking and new approaches for image watermarking, but none of them seem to be safe against attacks that use deep learning techniques. So our approach is to make the watermark physically indestructible, so that if you want to remove the watermark, you're going to destroy the whole image. And we're in the process of adding watermarks, not just to the Marlon Brando images, but we selected a bunch of other images that I will not discuss in detail, that are being right now watermarked in the non-destructive fashion. And we proved that physically, using any available enzyme right now, you cannot remove the watermarks and without destroying the image. And again, these watermarks may create little disturbances in the data, but we managed to clean them up with, in the Marlon Brando examples, with the machine learning procedures we had. And with this, I'm sorry I went a few minutes over time. I would like to thank you all for being here today. And I would like to thank the sponsors of this work, NSF, SRC, and DARPA. Thank you.
Thank you, Ogika, for this nice talk. So we go to the next speaker, and we will ask you questions at the end of.
Oh, okay, sure.
Session.
Okay. Yeah, I would have to probably leave a bit earlier, but I'll let you know if I have to leave a little bit before the end of the time.
Okay, let us know. Thanks.
Yeah, thank you.
Okay, now it's the presentation of Nimesh. So, Nimesh, can you share your screen, please?
Yes.
And yeah, if you can put full screen also. Yeah.
Okay.
Okay, so as you want. Thank you.
Mark, Turaj, thanks for inviting me to this talk. So we at HelixWorks, we are working on the technology to write data to DNA. And today I will be speaking about some of the breakthroughs that we think a lot about at HelixWorks, and we think that are needed to make DNA storage media feasible. So many scientists and engineers have demonstrated DNA storage systems over the last few years. And the amount of information that they have implemented in these systems has gradually increased, starting from just several kilobytes up to several hundreds of megabytes. And it's fair to say that many of these demonstrations would have not been possible without the advent of array-based DNA synthesis. Today, the dominant method for producing large quantities of DNA is array-based DNA synthesis. And when array-based synthesis had been commercialized in the 2010s, so it broke into a market that was predominantly dominated by another method called column-based synthesis. And while the chemistry used in both these manufacturing methods are the same, array-based synthesis represented like a major paradigm shift in the manufacturing machinery. Suddenly it became possible to make thousands of oligos for the same money as it was about 10 years ago. And so this new platform, it enabled increased automation, and most importantly, it also miniaturized the scale of synthesis. And that's why you were able to get these thousands of oligos for the price of one all of a sudden. And so, well, needless to say, it's widely accepted today that even with this state-of-the-art platform, it still falls well below the performance needs for DNA data storage. And so there are a handful of companies that service the market with array-based DNA synthesis. And because of the impetus to bring DNA data storage to commercialization, they have set off on further, with further goals to miniaturize this chemistry. And we should also note that there were refinements and improvements in the synthesis chemistry in the last few years. So previously it was possible to only generate oligos of 100 nucleotides in length, and it has risen up to up to 300 nucleotides today. And there are new biochemistries for writing DNA that are on the horizon and hitting the market. And so it's entirely possible to to envision that we'll have even longer oligos in the near future. So given these rapid developments in DNA synthesis space, many people think that the future of DNA storage will be defined by miniaturizing this chemistry even further. But the truth is that the writing chemistry matters more than people think. And so I want to submit to you some of the inherent limitations of the existing paradigm of synthesis chemistry. So for the purpose of simplicity, we can think of a DNA molecule as a string in a quaternary alphabet, ATGC. So in the current writing methods, so whether it is phosphoramidite or it is an enzymatic method, the DNA synthesizer creates a large number of DNA strands in parallel. And each strand is grown by one DNA base or one character at a time. And to append these bases to the strands, the synthesis machine performs a coupling cycle. And this machine iterates through these coupling cycles to add one base in one cycle. So since the DNA strand is a linear chain and has one of the four bases, A, T, G, and C, the specific arrangement or the sequence provides a certain amount of information. So we could say like the maximal amount of information that a single base can hold is up to 2 bits. And from the various implementations that we have seen, and depending on the encoding and decoding method used, I think it's fair to say that the capacity ranges anywhere from 1 to 1.6 bits per base. And therefore, for each coupling reaction, it is possible to write anywhere between 1 to 1.6 bits per cycle to the DNA strand. And we have to note that this coupling cycle reaction usually takes anywhere between 5 to 10 minutes, depending on the various chemistries or the various platforms. So I want to suggest that this method of writing data to DNA poses a higher risk, to make DNA media feasible, and we think that it's one that needs to be addressed. So at HelixWorks, we suggest one way to overcome this limitation is by first expanding the character set used for making the DNA string. So the benefit of expanding the character set is that we will be able to write more bits per coupling cycle to the DNA strand. And so we think there are like 2 possible ways to implement this. So one way to expand the character set is by using artificial nucleobases. And to our knowledge, like there are several artificial nucleobases that are commercially available, but typically they're very expensive. And so even if we build a set of characters that includes the natural nucleobases and the artificial nucleobases, we probably won't be expanding the set by more than maybe 8 times. And it's the uncertainty of the synthesis working and the cost is still extremely high. The other option is to use small oligomers and expand the character set. And so small oligomers, they can be coupled, they can be used in the coupling reactions, and this could be either an chemical ligation or it could also be an enzymatic ligation. But today I will not really be prescribing any one or the other method. I will just say that it is entirely in the realm of possibilities. And so these small oligomers would be designed such that they can be easily differentiated when sequenced. And these small oligomers would also be synthesized by the traditional phosphoramidite chemistry. And we think that it's actually very cheap to make these small oligomers using column-based synthesis. What we have found is that it is actually 5,000 times more cheaper than array-based synthesis when it is being measured purely in terms of yield. So if you're looking at the number of moles that we're getting, number of moles of DNA that we're getting with column-based synthesis, and the number of moles we're getting with array-based synthesis, and we compare the price, it's actually 5,000 times cheaper. So this would be suitable for a new chemistry. And the other thing to consider that we think that's important is, the cost of reading the data back from DNA is directly proportional to the number of reads that would be sequenced, right? So it's really important that the chemistry that we're using for these coupling reactions, it should be efficient enough that we're able to perform at least 200 coupling reactions per oligo. So to summarize, this new chemistry, it needs to have an increased or expanded set of characters, which could be in the form of small oligomers, so that it'll enable us to write more bits per coupling reaction. And it should also, the new chemistry should also be efficient enough that we are able to perform greater than 200 coupling reactions. And finally, the chemistry should, it should be amenable to be implemented on an array-based platform, so that we are able to miniaturize the synthesis chemistry to make it cost efficient. But that's all from me, folks. Thank you for having me. And if you're interested in working with HelixWorks or you have any questions, please feel free to drop it in the line. Thank you.
Okay, thank you, Nimesh. So next speaker is Marthe Collot from Imagen. So Marthe, I give you the floor.
Thank you. I would share my screen. Okay, and launch the presentation. Okay. So thank you for inviting me to talk about our technology for ambient temperature storage of DNA. We are Imagen. We are a small company based in Bordeaux, and we now have two sites, one lab research and development in Bordeaux and Biotech Platform in Paris. We are focused on the room temperature storage of biospecimen and DNA and RNA. So at the beginning of the company, we were created in 1999, and the storage approaches were based on freezers for DNA and RNA at -80 and -20. And it's still the case. And these have major drawbacks. They have high costs for electricity, maintenance, air conditioning, alarm and monitoring systems. It's very difficult to automate the storage and retrieval of the samples. And there's a high risk of loss or degradation of precious sample collection due to technical failures, power outage, or natural disasters. That's why we thought we should set a new procedure to store this specimen. And at the beginning of the company, we made key observations. And the first one is that atmosphere has deleterious effects on nucleic acids, and mostly is the moisture that enables chemical processes to occur, leading to cleavage or alteration of nucleic acids. And also it leads to DNA adsorptions or aggregations, and it prevents resuspension and recovery of the samples. And we also noticed that standard plasticware is not airtight, and atmospheric water can rapidly penetrate and alter the samples. So the breakthrough solutions that we developed is to completely remove water, oxygen, and ozone from the system, and to maintain these conditions throughout the life of the sample. That's why we developed these capsules. They are called DNA shells and RNA shells, and they are made of stainless steel. So we have a body, a metallic shell, containing a glass vial, and its cap, also a metallic cap, is laser sealed under argon and helium inside a glove box. And this ensures really good dehydration and prevention of uptake of water during the lifetime of the sample. The capsules are etched with 2D barcodes for full traceability, and they are put on racks conform to SBS standards for automation. So this technology has a lot of advantages. We have an easy access because it's room temperature, manual and automated storage. It's really easy. You save energy because you have no energy consumption when the encapsulation is performed. You don't need air conditioning or specific infrastructure. The tracking is very easy and secure because it's laser edged and it's duplication proof. You have a secure access because once a capsule is open, you know it. You cannot have accidental opening or cross-contamination. You save costs, obviously, because less space is required to store the sample, and there's no additional cost once the samples are encapsulated. And so it simplifies all the logistics for the storage and shipping of these samples. And of course, the integrity and stability of the sample is really nice. When you remove water, oxygen, and light, the stability is very good, and we will see that in a bit. So Imagen is a service company, but we also sell the instrument so that the customers are able to encapsulate the samples at their facilities, the consumables, and also the service at our service platform. We also develop a lot of accessories to be able to use the capsule in the lab. And recently, we also developed our own products based on the technology. For example, quality control reference samples for molecular diagnostics. Currently, we are able to store all of these samples, so nucleic acids, whether they are natural or synthetic, and coming from different sources in different formats. But we are also able to store nucleic acid sources, meaning the biospecimens that we put inside the mini capsules, so that we don't have to extract the nucleic acids before the storage, and we can extract at demand after the storage period. And we have also current development for viable microorganisms like viruses, bacteria, and reagents, PCR mixes, including enzymes and fluorophores. And these developments are always made through R&D contracts and partnerships. This technology can be applied to very different sectors and domains: biobanking of DNA, RNA, and biospecimen, molecular diagnostics, sequencing, biotechnology, precision medicine, forensics, and of course, data storage. One example of the application of the technology is the development of a kit for diagnostics. It's a kit containing reference material for the monitoring of every analysis that can dose the level of disease remaining for the leukemia. And it's RNA, and it's stable for 10 years. And we have a lot of laboratories that adapted this technology because it's really made their life easier because everything is at room temperature. And of course, we have other kits in development with natural and synthetic nucleic acids. Another example of the application of the technology is DNA data storage in DNA shell. It was a UNICEF Norway initiative to celebrate the 30-year anniversary of the United Nations Convention on the Rights of the Child. And the text was encoded by EMBL. The DNA was synthesized by Twist Bioscience. We encapsulated the DNA in Paris. And now this nice capsule is stored in Norway. So for us, it was, I think, the first time we were implied in the data storage sector. Now, our progress of the company is that we have an industrial service platform in Paris. We also sold 2 operational systems and also one abroad in Ivory Coast. We worked on the room temperature storage of microorganisms with the French Armament Procurement Agency. And now we have a lot of customers for services, but also for the kits of quality control for molecular diagnostics and all over the world. I'm going to present you the Biotech Platform that we set up. So we have one in Paris for the service, but we can also sell these platforms to our customers. So this is the workflow. We can receive biological samples or already extracted DNA and RNA samples. We perform extraction, aliquoting inside the mini capsules. The mini capsules inside a desiccator, where we remove the liquid water, and then the racks of capsules are put inside the glove box, where we have argon and helium at less than 1 ppm of water and oxygen, and that's where they terminate the desiccation of the molecules. And after that, we seal them with the laser, and we check for every leak of the capsule with a sealing control with mass spectrometry. The workflow is monitored permanently with a LIMS, and it allows the tracking of the samples, the operations, and the mini capsules. And if it's the service platform, we can send back the capsules to our customers. And we have a throughput of 5,000 mini capsules in 8 hours per day. Equipment that we developed. So we have 2 encapsulation stations with different throughputs, 1 leak test station, and also a storage station that I will show you later. And in the laboratories, we can have an easy use of the capsules because we developed readers and shell openers and shell replicas to be able to manipulate, open, and read the 2D codes. And you can replace 10 -80 freezers by 1 storage station, which contains 250,000 mini capsules in a 3 m² footprint, which is really nice for biobanking also. Once the DNA is dried inside the mini capsule, it's really easy to recover it. You can open the mini capsule with a shell opener. You just add water or buffer. You wait maybe 10 minutes, and there are no further chemical steps. You mix the solution by pipetting, and the DNA is ready for use. And we have a quantitative recovery from picograms to milligrams of DNA. Now I'm going to show you some scientific studies, and the first one is when we started, we studied the DNA damage sites and effects, and we studied the attacks of water, oxygen, light, ozone in the literature, and they lead to either chain break directly or base loss or alteration of bases or phosphate, and a lot of these alterations end with chain break, and depending on the environment, you can also have cross-links or aggregations. But for our studies, for degradation studies, it was nice to see that chain break is the main deleterious event that we can follow very easily with different techniques that I won't detail. And the main event is depyrination followed by chain break. And the main degradation factors are water. Water is really key, but also oxygen, ozone, and other atmospheric pollutants, and contaminants and co-extractants are also an influence. And at the very beginning, we wanted to see if tubes and vials could be found, tubes and vials that are watertight. And so we compared different kinds of containers, vials, creamed vials, different types of tubes in plastic tubes, O-ring tubes with a silicone joint, closed tubes that were enclosed in bottles. We put water sorbent inside these containers, and we followed the water uptake through time. And we saw that there were no container was able to keep water away. So we really needed in hermetic containers, and that's why we found the best closure, the best system to close and to remove and to stay away from water was to laser yield the metal. So that's why we developed the DNA shell and RNA shell capsules. Our first publication was in Nucleic Acid Research, and this paper was like our founding paper, and it's the first time that an Arrhenius equation was established for determining degradation rates at room temperature of solid state DNA. And the main conclusion was that DNA, if it's kept away from degradation factors, light, ozone, oxygen, moisture, is really stable. We found that we had 1 to 40 cuts per 100,000 nucleotides per century. And so this paper was has been extensively cited, and we're very proud of this paper, this first paper. But at this time, we didn't have the capsules to prove our point. So when the technology was mature and we were able to have the capsules, we wanted to confirm that the capsule was able to protect DNA. So we conducted a kinetic at room temperature for 32 weeks, and we compared 4 conditions. So the capsules, but also DNA that was dried in tubes and left at room temperature without any other protection. So the DNA is in contact with the air and the water and the air, because we had relative humidity of 50%. And we compared this with the use of matrices, Trehalose matrix and a commercial matrix, to see if they were able to bring some protection to DNA. And what we saw is that we were able to recover DNA with the matrices, but DNA was rapidly degraded. So when you have no protection at all, you have aggregation and rapid degradation. Matrixes can bring some protection, meaning that you can recover the sample, but it's completely degraded after 8 months. And we prolonged this kinetics, and you can see that in the capsules, the degradation is very, very limited. So we were able to show that the DNA shell offers an efficient protection to DNA at room temperature. We applied the same technique for RNA. The process is a little bit different, but we also established an RNA model, and RNA is really stable also. And we did the same with DNA inside the biospecimen. So it's buffy coat and cells that were lysed and put inside the mini capsule, and we also established that it is very stable. And we are recently we've been evaluated by an external team from Microsoft Research, University of Washington, and ETH in Zurich. And they performed this evaluation on 29,000 DNA sequences. And they evaluated qPCR and sequencing. And here you can see that all these conditions were compared, and here we have the capsules where the DNA degrades very slowly. So they prove that this is really the best way to store DNA in the long term at room temperature, to be really able to stay away from water from the atmosphere. And these are our last results that are about to be submitted. So it's the RNAs in the DNA shells, because after the nucleic acid research paper, we didn't do this again, so we needed to do it. So we use here a 2-size amplicant qPCR system to determine the degradation rates at each temperature, and this allows the extrapolation. And we found that we have 1 cut per century every 100,000 nucleotides, which means that we have a half-life of 38,000 years for 150 nucleotide long fragments. And we compared these results with different other procedures and methods. And you can see that the capsule is really the only way to have a physical protection of DNA to protect it from water. So to conclude, you can see that we have a technical superiority concerning DNA stability. It's the only procedure that can really protect the water uptake back from the atmosphere. This technology is mature, it's extensively validated, and it's already industrialized. And so it's already usable, and it's already used for DNA data storage, for example, for cold data and also for large amounts of DNA. And thank you for your attention. You can answer the questions at the end. Thank you.
Thank you, Marthe, for this nice presentation. So, okay, there are some questions in the chat, but the questions are, all of them are from Touraj. I don't know, Touraj, if you want to ask them orally, but I guess they were related to the presentation of Ogika Milenkovic, and I guess she is no more connected right now.